I'm going to spend the next little bit of time discussing the Immunome Browser, which is a tool that we have, and then do a live demonstration of it, and hopefully all goes well. The Immunome Browser is a unique tool that we have that is a tool that's not available in the analysis resource, and you'll understand why when I show you in a minute. It's actually integrated into the search interface. So this is something you access on the results page from the query, because it's very specific to the query that you're running. And it's very specific to the antigen that it is mapping to, all right? And that'll become clear in a minute. So what is the Immunome Browser? Well, it's defining the immunome for that particular set of circumstances that you put the input for your query. The totality of immune reactivities at the organism level. So that sounds kind of vague, but you get the idea. It's all the responses to that particular antigen in that particular context. Why do we have it? What is it for? A very frequent question of people coming to the database and coming to us and talking to us about epitopes is, what are the immunodominant epitopes, right? That's what everybody's interested in. And it's not as straightforward to get at that as one might think, because it depends on the person or the host, because it's different for different species, it's different in different contexts, people from different places, different viruses, different strains of viruses. You get the idea. It's not as straightforward as you think. So input criteria, so the circumstances that you're interested in, are criteria that you can put into your query to get at that question. What is an immunodominant response for that set of circumstances? And that's what this allows you to do. How did we do this? Well, the best way to do this is to use response frequency data. So very often, a paper will tell you the number of people tested and the number of people responded out of the total tested. All right? Not all papers do this, and it's more frequent in human data than it is in mouse data, as you might expect. But when that does occur, we can use that information to give you an idea of prominence of a response to a particular region on a particular antigen. All right? And this works by plotting the response frequency for each epitope residue along the length of a reference antigen or a reference proteome, right? It's designed to let you visualize and identify a subset of good, so to speak, good epitopes or great epitopes among the mass of data that's in there using specific criteria, so user-designed criteria, right? And I'll show you how this works. A really good example of this is HCV. Those of you who are not familiar with HCV, it's a virus with a genomic polypeptide. So you have a polypeptide, so all the proteins are encoded therein. That's about 3,000 amino acids long. If you do a search on HCV in the IEDB, you get 4,200 epitopes. Now, obviously, not every single residue in that polypeptide is an epitope, right? That seems ridiculous. But what this reflects is not bad research. This just reflects iterative research. So many labs have worked on this, many different assays, many different species have been covered. So lack of a better way of putting it, the HCV is very well covered, so to speak, okay? So how do you get at the choice epitopes among this forest of epitopes that you'll see? And the way you do it is with your query input. So you can be very broad or you can be very stringent depending on what you're doing. You can look at just human data, just neutralizing assays, just mouse data, CTL assays, just discontinuous epitopes. You get the idea. So as stringent as you want to be, and this gets at how do you get at good data, what you consider good data versus bad data. Well, it's up to you. You can put in very stringent conditions and look at it that way. So I'm going to show you what that looks like. So from the home page here, you would type in HCV. The autocomplete will come up, and you select the one you're interested in and click search. Then here on the results page, which I've just clipped out a little snippet of, I'm not showing you the whole thing, I've clicked on the antigen tab because you're plotting the epitopes to a reference antigen, so you start from the antigen tab. And so you come down here, if the mouse will work, and you pick the genomic polyprotein here and click on the Immunome Browser icon, and this will map the epitopes from that query to the reference proteome in this case, because it's a polyprotein from a virus. And this is what it looks like. So I'll walk you through this. These are the two graphs that you'll see. The tool generates an upper graph that shows you the confidence interval, the upper and lower bounds of the confidence interval of the response frequency score. So number tested and number responsive, okay? And that's here. Below that, so along this axis you see is the residues of the polyprotein. So I've overlaid here this graphic of the HCV polyprotein, so from the N-terminus, the core protein at the N-terminus, to the non-structural 5B protein at the C-terminus. So you get an idea of what this axis is representing. It's representing all of the proteins. You can and I'll show you this in a bit, you can just look at core, just look at E, you don't have to look at all of it. You can shrink down and zoom in, and there's different ways you can do that. But that's a functionality for that. The lower graph, so as you can see, there's different regions. The core protein in this case, we know because we studied this and we published a meta-analysis on this, is a hotbed of antibody response because it's an exposed protein. And then there's other areas of the protein that have hotspots, for lack of a better way of putting it, hotspots of reactivity down in these other non-structural proteins. Down below, the graph shows assay counts. And what we mean by that is showing you this iterative that there have been literally hundreds of assays captured in the IEDB describing the epitopes defined at these locations. It also shows you whether or not there were negative assays. So this is nice because at a glance, this graph is showing a very well-covered organism, but very often you'll run one of these and you'll see blank spots. So you don't know whether the blank spot means it was tested in negative or whether it was tested or not tested at all. So it gives you that kind of idea. So that's why the assay counts are very important, because they give you that information. So this shows you overall coverage, and this shows you actual response frequency. And then you can use refinement of your input criteria to kind of get at less noise, so to speak, depending on whether or not you've pinpointed the response types or response that you've been interested in. Then if you scroll down, and I'll demonstrate this when I show you the live example, as you scroll down, you'll get the actual data table that provides the response frequency. So starting over here on the left, this column is the epitope ID. If you were to click on the blue, it will take you into the record, as we showed you earlier with the question for cyanobacteria. It will take you into the actual record so you can see the details for that particular epitope from this row. This is the epitope sequence here. Then it's aligned by, in this case, position number from 1 all the way down to 3,000 if you were to scroll down all the way. This percent identity is the percent identity of that epitope with the reference proteome that it's being plotted to. Then here you get the raw data, the number of tested subjects, the number of respondents, so you actually can see 14 out of 14, 0 out of 3, and then here these two columns summarize positive and negative assays. And then finally, over here on the far right is the response frequency score from 0 to 1, 1 being best. All right? So what I'm going to do now is I'm going to take you through that example live. All right, and if you want to follow along, that's good. All right, so I'm just over here on the home page. So from the home page, I'm going to type in HCV. Autocomplete's going to come up with choices, and I'll select that. And then I'm going to hit search. And then again, along the top, you have the 4 tabs summarizing the data for that particular query. I'm going to click on the antigen tab, and then I'm going to come down to the genomic polyprotein, and I'm going to click on the icon for the Immunome Browser. So now that it finally came up, so this is a live version of the HCV. Remember, the input on the home page query interface was HCV, and then I selected the genomic polyprotein and hit go on the Immunome Browser icon.
Can you tell us how you got there again?
Yeah, sure, I can back up. All right, from the home page, UCV, select, and hit search. And then to the assay, or sorry, to the antigen tab from which the Immunome Browser works. I've selected the genomic polyprotein. If this was another, say a bacterial protein, you'd get a list of different individual proteins and you could click on those individual ones. This is how these viruses work because there's a genomic polyprotein. But we can show you examples of individual proteins later. Well, I actually am. I'm going to show you an insulin example later. Whoops, sorry. Yes, sorry. Just kidding. Okay, so up here you see the upper graph, which is a reflection of the pattern of responses based on response frequency score. So total number of people responded over tested. So the higher the number, the more people responded to that particular region. Again, this is a core protein over here, and this is NS5 over here. Some functionality I'll show you. If you have this mouse over here, so you can go to an individual residue, and it tells you the range of the confidence interval, so the upper and lower bounds, so 0.04 up to 0.08. In this case up here. The tighter these two colors are together, for lack of a better way of saying it, the more confident you are in the response, the data that's there. The other neat feature is you can quickly do this. You can narrow it down to a particular region. And then it'll show you that region. So now we're at 528 to 1055. You can also come down here and do the same thing. You can input the particular protein. If you know the residues, the residue positions for your particular protein, you can enter those in and zoom in that way. And then you can hit reset if you want to go back to the broader thing. Again, down here on this lower graph, it does the same thing. You can do the mouse over and it shows you number of assays, in this case, it's the blue and the red are showing the number of positive in blue and number of negative in red for a particular region. So you can see well-covered regions. And sometimes the number of assays is quite high in the hundreds, and sometimes it's quite low. It just depends on a particular epitope. Sometimes they're well-studied, sometimes there's not a lot of data. It just depends on the particular thing you're studying, on the relative abundance. Up here, this is a feature that I love because I used to have to do screenshots to do this. You can download this graph. So if you want to publish this in a paper, the data that you've generated here, you can make this a PDF or a JPEG and have that image for your paper, which is a really nice thing. You can also print it and save it. So and annotate it in different ways. Let's click on that.
A question?
Yeah.
If you go to this around 1650, where you have a number of positive and negative assay counts.
Down here.
Perhaps my understanding of the confidence bounds is wrong, but should you expect to see a wider confidence interval at that?
Tell me again the positions you're referring to.
Around 1650, 1700.
1650?
Yeah.
On the upper?
Yeah,
Here?
so there's, yeah.
Right here.
So there's seemingly not very much difference between the upper and lower bound.
Yes.
I don't know if it's meant to reflect the disparity between positive and negative assays.
No, for the positive assays, if I'm going to see if I say this correctly, for the positive assays from which this data is coming from, the upper one is coming from, the fact that the two pinks are very narrow means that there was very low range in the confidence interval, which means that there was more confidence that it was accurate. Bjorn can say it better than me. But so we can go 16. Oh, I'm sorry. It's not still in annotation mode. Word to the wise, hit cancel on annotation mode if you're in it. All right, so now we're here. So it did it. So here we're now 16 to 1650. And yeah, we zoomed there. Maybe I should zoom out a little bit. Yeah, so I think it was this area here that you were talking about. Does that look right? Yeah. So here, yeah. So there's a very good correlation between the two pinks there. How many negative assays? There are for the same spot, 25. So, I'm sorry?
He's saying that because there's so many negative assays, he would have expected the confidence interval to be bigger.
No, I don't know that would be true. Bjorn? No. What the lower graph reflects is clearly this region has been heavily studied. And because I put in no other selective criteria other than HCV, the negative assays could have been mouse assays and so there's different species here, there's different assays here, there's both B and T here. That I'm actually glad you said that because it brings up there's no specific selection criteria. So if I were to back up and refine this, rerun this with different criteria saying just T-cell assays in humans, this would look very different. So, and then if you do that, you can go back and then look at the same region and see. And when you do that, this tab here stays there. So when you run a new one, you'll get a new tab so you can flip back and forth and see them and compare them.
So the top graph, it's also capturing all data.
It's capturing all of the data for the query you ran.
Yeah.
Yeah.
So
in this case, it's
human, mouse.
broad. Yeah. So this is all 4,000 epitopes from all assays, all species. Yeah. Yeah. Okay. And let's see. Let's scroll down. So this is where I was talking about scrolling down. So this is a data table here. And again, if you click on these blue, you can go down into detail into the, this is a summary for this particular epitope. And in this case, the T-cell assays here. So if you click here, you'll go into the T-cell assays, similarly to if you had run a search. So you can quickly look at that, but then you can go back to the table. So you can tab back to the table. If you click on a row, the table will highlight what epitope you're clicking on from that row. Other features to highlight. One thing that's kind of neat is that I like to do is I'll download this in Excel. Fingers crossed it doesn't take a long time. Because you can use Excel to sort on the response frequency. So you can sort on the high response frequencies and look at those individual epitopes. And I'll show you an example of this when we do the insulin query that I'm going to do in a few moments or after lunch. So here's what it looks like. And so just to familiarize you with this. So these are all the same columns from the table, from the data table you just saw. You can get yourself familiarized with how it's set up. In this case, I'm going to highlight the response frequency. So then I'm going to sort this, large to small. And you can see, so you can go down and see that there's many responses that are one. Here we go. So you get an idea that you can quickly look at these and you can zero in on different ones. And because you have right here the raw data, you can see the response frequency here, and you can see the individual epitope. And so you can get an idea of, because this is the actual calculated RF score, whereas the graph is a average of each residue position for all the data that's been plotted. Let me go ahead and put that down. Yeah.
So if I want to see the same Immunome Browser but for a specific assay, I can add that to the Immunome Browser and make the search.
Yeah, so say we, so this was the search that so I'm just going back, I'm tabbing from the display that I've been currently working on, and I'm going back to the IEDB results page. And from here, I can come down here and look up tetramer assays. So, and then refine that query there. So I still have the original one up that shows all. Again, I'm going to repeat selecting from this query the data that's mapped to this particular polypeptide. And so it should look a lot different because it's very refined to just a small number of assays. So you can see it looks very different, all right? And so from this, well, let me go back and reset this. From this being all to this just being a subset of that data. So what the nice thing is you'll have this, as you refine it further and further, it's just a subset of the larger data that you do. And that reminds me to remind you or encourage you that when you come to the IEDB, depending on what you're looking at, start your searches broadly, because a lot of times people will type in very specific things, they won't find something, then they get frustrated. But very often there's a tremendous amount of data in there. It's just a matter of it wasn't as specific as the assumption goes. The famous example I give is a researcher who came up to me about B-cell assays and ELISAs, B-cell ELISA data, and he assumed that all of them were quantitative. And I said, no, we call them positive or negative because that's what the paper showed. And he was incredulous. He couldn't believe it. I said, no, people don't, because the actual OD between labs varies, so we don't capture that. I'm bringing that up is to point out there are some certain assumptions that we make about the data in general. They don't always hold true. So go more general, and then you can look either on the spreadsheet if you download that for your specific instances, or once you see a body of data, refine your search until you find what you're interested in.
In the data table at the bottom of this,
Yeah.
Can you describe the difference between a subject responding and an assay being positive?
Yeah, in an assay in which the subject responded is counted as a positive assay. All right.
If any one subject.
Yeah, any one subject. But there are also assays that are positive assays in which they do not report a response frequency, but that still counts as a positive assay. Right. And similarly, yeah, go ahead.
So just in the IEDB, we call it one assay if they have a group of 25 people that they test. You could say that's one assay, which is kind of stupid. So it's one group of experiments, essentially. So then the overall experiment would be positive if at least one person responds within that. So.
Yeah, so an ELISA performed testing the sera of 25 people is referred to as one assay. That ELISA is the one assay. It gets down to our jargon that we use, that we must use in order to describe this for the purposes of the database. But sometimes it can be confusing.
Thank you.
Sure, yeah. Any other? Yeah.
In the identity table, you have sometimes different percentage from 100.
Yeah.
And I believe the minimum was 70. But in that same column, I can find for a different protein values up to 30, 20.
Yeah,
That depends of what? That depends on what of that?
So this column here, percent identity, is the identity, the match of this epitope sequence to the reference proteome that it's being mapped to. In many cases, it's 100%, but often it's not 100%. And I think the lowest, what is the lowest? Is it 70 or 60? I don't remember.
The lowest I can find is 30 something.
Pardon me?
34. In the one I'm looking, I found 34.
34?
Yes.
Ooh. Okay.
So this is a very different percentage than what we looked at before in the query. So this is, as Carrie was saying, we have a reference protein for HCV, And if we can identify one specific region where this peptide matches to, that's what we would report. And so it depends more on the number of matches, and is there any other region in the protein where it could have also been? If we can say with certainty where it goes, then we report it. So it's not a straightforward threshold of % similarity.
Yeah, the idea being that we're trying to represent visually along a representative antigen all of the data. And so to the extent that we can do that, that's what it's reflecting.
I'm sorry if you may have answered this already. I may have missed it. On the table header, next to results returned and displayed, there's a toggle box that says display graph residue positions.
Yes.
I've played around with that before. It doesn't seem to do anything.
It's not functional right now.
Oh, well, that would be why.
It's a work in progress, but it's not functional right now. This is a 3.0 version. There was an original 2.0 version of the. No, doesn't matter. Anyways, yeah, it's not.
Okay, thank you.
We won't get into the nasty details. All right. Any other questions before we do lunch? All right. Thank you.